So this is the week 11th assignment session, almost your second last week. So in this session we are going to discuss few questions related to fermentation of other different products. So let's start. First question is, if there is any issue in the voice, then please let me know. First question is, which of the following pesticide can be used to treat crown gall disease in plants? So in this question they are simply asking about the pesticide name that used for the crown gall disease. So what are the options? Options are, first is parasitoids, second is dendroidemus, third is agrocin, and fourth is phenazine. So for this disease, which pesticide is used? Anyone want to answer this? You must know this. Okay, for this, someone answered Puja. Yeah, see. Okay. So, yeah, correct answer. This agrocin is used for this crown gall disease. You have, for this question, answer is C. You have studied different types of pesticides, either microbial pesticide, biopesticides, and different other pesticides. Then you must know the example of each type. That is really important, and especially if they are related to any particular disease that is very common in plants. So then you should know the particular example also. Like this is the agrocin, and this agrocin is mainly from the this source, this agrobacterium radiobacter. This is the microorganism that is used for this agrocin. Okay. So now coming to our second question. All questions are really simple. Like you can easily know. Like if you have read the your lecture material once, then even you can answer all these questions. So this is the second question. They are asking what are the plant incorporated protectants or PIPs, like they are calling it in a short form this. So the name itself telling you what is this, like what they are asking for, plant incorporated protectant. So from the name itself you can identify what are these protectants means, that can protect the plant from, or incorporated, it means they are already present in the plant. Suppose this is your plant, so this plant already contain these molecules or components that can help in protection. So the name itself define this. So let's now read the options. First is they are derived from fungal source. No, they are, like even they, the question itself saying that plant incorporated, so it means they are already present in the plant, so they can't be from the fungal source. Then second option is they are antibiotics which colonize host roots. No, this is also like from outer source they are talking. And the third thing is they are pesticidal substance that plant produce to kill pest. So this is the correct one, that these are the pesticidal substance that are produced by the plant to kill pest. And this is the thing that we usually do in the genetic engineered plants. What we do, we just incorporate some genes into the plants so that will make the plant to either kill the pest or make it resistive. So for this question, answer is C. So this is the next question. Match column A with its correct option in column B. Okay, in column A, the four different this microbial names are present, and these are the functions they are talking about. So in this, they are mainly talking about these are the like pesticides that we take. So these are the examples of like microbial pesticides. So what are the functions? You have to match that, or how they will help. So from these four, this one is really like very common that we use baculovirus. So this is mainly used for, anyone want to answer this? This specific, this fourth one baculovirus, this is used for the control of what? Okay, aphids. No, no, Satvika, this is not correct. Actually, this baculovirus, this baculovirus is mainly used for this control Lepidopterus and Hymenopterus pest. For this, we use this baculovirus. Okay, now let's mark the others also. First one they are asking is Metarhizium anisophila. So this is mainly used for the this spital bugs or rhinoceros beetles. Okay. Then the second one is Beauveria bassinia. This is mainly used for the control of this potato beetle, Colorado potato beetle. And this Verticillium lexina, this is used for the aphids and whiteflies. So in this order they are used. Like you only want to know, you must know the function of this, like from where or how they are using. Only this much. You don't know to go in much detail about all these things. So for this, like if you mark for first one, this is D. Yeah. So this is the C option that is correct for this question. This baculovirus is used for Lepidopterus and Hymenopterus, and this Verticillium lacina, this is used for aphids and whiteflies. And even apart from these four, other examples are also mentioned. That you must read them once at least to know the function. In your lecture material, these examples and other examples are also mentioned with their functions, so at least look them at least once. So for this question, answer is C. Now this is really very simple question. They are asking which one is the example of bioplastic, and what are the options? First one is monolactic acid, second is polylactic acid, third one is polylactose acid, and fourth one is monolactose acid. So anyone answer this? Because this answer you should
Lactic
tell.
acid.
Sorry?
Polylactic acid.
Yeah, polylactic acid, that is correct. This polylactic acid is the example of bioplastic. Means this is used, like this can be biodegradable and this is like recyclable. And this is taken from the uh, biological source. So this is this polylactic acid that we are using in many different ways. This is the example of polylactic acid. So for this question, answer is B. Okay, this is the next question. Prolamine protein found in, they are asking the source that can be used to produce in. So anyone want to answer this? Okay, Satvika maize. Yeah, this is correct. That this prolamine protein is mainly found in maize that can be used to produce zane. Can anyone know why this zane is used or what is the function of this zane protein? Correct answer is this B, that prolamine protein that is present in maize is used to produce zane. But why why we want this zane? Okay, what happened? This zane is mainly used in food industry, food or packaging industry specifically. Main function of this zane protein is that it either lock or it will not allow moisture. So when moisture is not allowed, that is major problem in the food industry. If the food containing get the moisture, it will get rotten. So this help in preventing this. So this is mainly used in the food and packaging industry. Okay, so for this question, answer is B. Okay, this is the next question, and this is that we already discussed in the last to last question, that what are the advantages of using polylactic acid, that why we are using this as a bioplastic, because this is biodegradable, correct, biocompatible, that is also correct, and this environmental friendly. Okay, so for this advantages of using PLA is all of the above. Can anyone tell me the difference between these two words, that what is biodegradable and what is biocompatible? Anyone want to answer this? Or what do you guys understand the by these two words? That if
Biocompatible is it suits to any biosystem.
okay,
Like biodegradable means it consumes like it disappears.
it is a
It consumes, it disappears. Like degrades
yeah, it can be degraded using any biological
system.
system. Yeah, correct. Yeah, this is correct. That this biocompatible means that this is compatible or it can like it will not harm the biological system if it being with the biological system, and this is that it can be degraded by any biological system. So this is correct that you have answered. So you must know the difference between these type of words. So for this question, answer is D. So this is our next question. What they are asking, the vaccine for hepatitis B virus is produced by expressing the HBV surface antigen using the expression system of, okay, they are asking about from which microorganism we are using the system for this expression hepatitis B virus vaccine, especially marking this antigen. So what are the options? First one is Saccharomyces cerevisiae, second is Synechochitis species. The third one is E. coli, and fourth one is Pseudomonas. So this hepatitis B virus is produced in which expression system? Okay, Pooja. A. Yeah, this is correct. That for this hepatitis B vaccine, this is Saccharomyces cerevisiae is used as an expression system. You all have studied about like the production of vaccine, especially we are talking here about hepatitis B vaccine. So what happened? They will suppose this is targeted for the production of this vaccine for this hepatitis B virus. We target what? HPV surface antigen. So this is mainly produced by the recombinant technology. So what happened? They require a system, or you can say microorganism, to mimic or to produce that protein specific to this. So for this we require the specific microorganism, and this is really important to choose a particular microorganism for the production of any protein or any particular antigen, because this is really important. As suppose you are producing any protein A, and you have chosen a microorganism, suppose this is a microorganism that you have chosen, and if there are interference, if its system of B interfere with this A protein in any kind of thing, either it will react with A protein or it degrade the A protein, the enzymes present in B, or it can change its structure of A through any other means. So all these conditions need to be fulfilled, that this A protein should be produced in B as it is that we required, because only after that this A protein is useful to us. If it undergo any changes, it won't remain the same, and you will not be able to produce the particular protein that you want. So the selection of this microorganism is really important in this vaccine production. Although in all the process of fermentation that you are studying from the day one, it is important to choose a microorganism, because that chooses or major factors depend upon the microorganism. Suppose if you are working in an industry, so the amount that is producing, if it's cost effective or not, all these things are depending upon microorganism. Same thing is with the vaccine, that if you want a particular protein, that you must know the system of every microorganism. So for this, selection of microorganism is also a critical part, so that its machinery did not interfere with the particular protein that you require. So for this for this hepatitis B vaccine, we usually use this Saccharomyces cerevisiae. Even other microorganisms can be used, but we need the very effective, like very close to the humans, because when we use the vaccine for the humans, then it will also not that some genes of this will come to there, and for interference also. So all the things need to be taken care. So for this question, answer is A. Now this is the next question. The first vaccine produced against HPV by Shanta Biotechnics Private Limited, India is. So they are asking about the name that we given to the first HPV vaccine.
Naturally, Shanvac B.
Shenvac B. No, they are asking about the first vaccine, and that is against HPV. That is Guni. This is the name they have given to this first HPV vaccine.
Okay.
Yeah. And in which year they produced this HPV vaccine in India? It is around 1997. This first vaccine is produced. And also, India is the fifth country at the time to produce this vaccine. So, for this question, answer is B. Okay, now coming to the next question. The recommended dose of hepatitis B vaccine is, the first option is 2 shots over a 24 months period. Second is single shot after birth. 3 or 4 shots over a 6 months period. 3 or 4 shots over a 24 months period. So this anyone want to answer this? Okay, this hepatitis B vaccine is mainly like recommended for a for a period of 6 months by in 3 to 4 shots. That depends upon the like age, gender, and other things. But usually it will give, if you anyone has like a very little baby in their family, then you must know that when the baby is born, they have a like card in which all the vaccines are mentioned. In that also they have mentioned that doses and the number of doses that is required and that is not required. Like for some vaccine only single dose is required. So that, so for this, from there also you can see what are the vaccines that you have used only once and how many shots you have taken for the particular vaccine. Satvika D. No, Satvika, over a 6 month period, this is given. Hepatitis B vaccine. So for this question, answer is C. Okay, this is your next question. They are asking about the human insulin composition. Because you have studied about the insulin. So what happened initially when insulin is started producing by companies or industries, it will be the source of insulin is animal source. Initially they are taken from. But with the advancement they have found out that this can be produced by the recombinant technology in the microorganism. Similarly means expression system we can produce this structure. This is the structure of insulin. Okay. This pink one is A, like chain, A chain, and this purple one is B chain. Okay, in A chain there are 21 amino acids. In B chain there are 30 amino acids. And these two, in this you can see that SS, this SS bond is present, means this is the disulfide bond. So this is the structure of insulin. So when initially we started producing this insulin using microorganisms, or when first it is tried to develop this insulin in a microorganism system, so what happened, what is the issue with this, that if we produce we have to produce A and B chains separately. Then after the production of this both A and B chains, then this disulfide bond that we have to done in a, not in the microorganism, in the outer, or you can say from uh, no, this is not, this is artificially that we add these chains. So bonding between these two chains will be done from the outer surface, from the, and that is not occur in a single organism. What happen, A is produced by differently, B is produced differently, then we combine these chains by providing, or from the, by making them to make these disulfide bonds. So this is the major issue with this insulin production. So for this question, answer is C, because they are asking the composition of human insulin. So human insulin is composed of two peptide chains. This A having 21 amino acids, B having 30 amino acids, and these are linked through the disulfide bonds, not the peptide bonds. This is important, that they are bind through the disulfide bond. So for this question, answer is C. Although it is not required for you to have detailed study about the insulin further, but if you will go through the structure of the insulin, then it is made up of three different chains. These two chains, and this is the one pre chain is also present in the structure. From this two chain structure is formed. So this is the finally insulin that we use, or that is produced in the body. Okay. So that's why this is important if they are asking for human insulin, and if they ask the precursor of this structure, then that is usually made up of three chains. So for this question, answer is C. Now this is the next question, in which they are asking the microorganism is used by Novo Nordisk to produce insulin. So what is this Novo Nordisk? This is a, this is the name of a company. So this is the company that produce insulin. So they are asking which microorganisms they have used. First one is bacterial cells. Second option is yeast cells. Third is animal cells, and fourth one is microalgal cell. So this, from this you can see this Novo Nordisk use the yeast cell for the insulin production, and this Eli Lilly, this is also the another company that uses E. coli, and this is the first company that start the insulin production. And these two are the major companies that produce the insulin. So this is the difference in their production, that they uses different type of microorganisms. So for this question, answer is B. So for this question, answer is B. Now coming to the next question. This is the conversion of atmospheric nitrogen to ammonia or nitrogenous compound is known as. Pooja, you answered this. Nitrification. Okay. Anyone want to answer this question? The conversion of atmospheric nitrogen to ammonia or nitrogenous compound is known as.
Nitrification.
Okay. Okay, I'll show you something. Okay, we'll discuss this question later, like after other questions. Okay. So this is our next question, that the following is our example of microorganism used for phosphate fertilizer. So what are the examples what they have given? First one is Pseudomonas, second is Aspergillus, third one is Mycorrhiza, and fourth one is all of the above. So for this phosphate fertilizer, what are the microorganisms that they have used, or that we can use any microorganism? Okay, this Pseudomonas that we know, that we normally use. Second one is Aspergillus. Third one is Mycorrhiza, and fourth one is all of the above. So these all three microorganisms can be used for this phosphate fertilizer. So for this question, okay, you can see from this, in this table they have given this nitrogen fixing biofertilizer. What are the examples? This Rhizobium, Bradyrhizobium, Azospirillum, Astobacter. Then this PSB, phosphorus solubilizing biofertilizers. For this Bacillus, Pseudomonas, and Aspergillus. Or phosphate mobilizing mycorrhiza, and fourth one is plant growth promoting biofertilizer. This is Pseudomonas. So especially for this phosphate fertilizer. All these three microorganisms can be used. For this question, answer is D. Uh, just a minute. So, from this you can see that Pseudomonas and Aspergillus, these are used in phosphorus solubilizing fertilizer, and this mycorrhiza is used in a mobilizing biofertilizer. So then, so you must know like how these two different, although they did a similar type of work, that they provide the phosphate to the plant but in a different pathway. Like in phosphorus solubilizing biofertilizer, the pathway of their working is different, and for this phosphate mobilizing biofertilizer, the pathway is different, but at the end their work is same, to provide the phosphate to the plants. Okay, so all these are used as a phosphate fertilizer, whatever their pathway is, but they are used for phosphate fertilizer. So both these examples are a part of this phosphate fertilizer. Okay, so for this question, answer is D. Now coming to the next question. This one is, which of the following microorganisms can fix atmospheric nitrogen? So what are the options? Options are, first one is nostoc, second is chlorella, third is chemidomonas, and D is all of the above. So for nitrogen fixation, which microorganism is used? Satvika, you answered for this question, question number 14. Let's see. Previous question. Okay. So for this question, what is the answer? This nostoc is used for the atmosphere, for this nitrogen fixation. Okay. So for this question, answer is A. This is your last question for this assignment. So they are asking what are the steps involved in anaerobic digestion process. So there are 4 steps in anaerobic digestion, but they are asking in a particular order. Like what is the first step and then further. So what are the options given? This methanogenesis, acetogenesis, hydrolysis, acidogenesis. So you have to mark like what is the first step. C. Yeah, both both of you are correct. First step is hydrolysis. Okay, let's discuss it here. So we are talking about the anaerobic process. They are asking what are the steps involved in anaerobic digestion. Okay, we are going to discuss it in a little bit detail. So what happens in any type of digestion process, whether it is anaerobic or aerobic, what happens? You have the larger molecules. Okay, that are degraded by different process into smaller. Like this is a very brief or like layman idea of any digestion process, that the uh, things or molecules are present in a complex form, or you can say that here in a larger molecule, complex form to simplified or very small. So this is the four steps of the anaerobic digestion. First one is hydrolysis, second is acidogenesis, third is acetogenesis, and fourth one is methanogenesis. What happens when you have the complex biopolymers, like suppose proteins or polysaccharides you have, then by the help of this bacteria or the microorganism that you are using, it will break down this protein or polysaccharides in a little bit smaller, like monomers and oligomers. Suppose this polysaccharides form the sugars, from this amino acids and form some peptides form. Suppose this protein contain a chain, so suppose initially it will degrade into single chain. So these peptides also form. So this is the process called as hydrolysis. Then after the hydrolysis, what happened? Mainly use in all these, the function of this microorganism is important. What happened is sugar and amino acids, this will break down into smaller form, or especially in the this volatile organic acids. This type of form is majorly formed in this, in the second phase. This will break down into mainly in a acidic form. Then this acidic form is converted into acetate form, and this acetate form finally form the this methane and CO2, because they can directly be removed or used. Means you have to like very simple process, that you have first you have to degrade in a smaller component, and you have to degrade it till smaller that it can be further used. It can be like it cannot be degraded further, and it can be removed or used easily from that process. So at the end stage you must require that it will convert into CO2 and CH4 form, mainly methane. So for this you need, methane is formed from this acetate, so you need a acetogenesis state. Then acetogenesis, some compounds form this acetate from the acid. So that's why this acidogenesis step is required, and this hydrolysis is required to form a little bit smaller components to break down the complex form. Okay. So for this question, answer is C. That first is hydrolysis, second is acidogenesis, third is acetogenesis, and fourth is methanogenesis. Means it will break down into smaller component, then acidic form, then it will form the acetate, and at the end mainly methane. So in this series, this anaerobic digestion will occur. So for this question, answer is C. So now let's go back to our question that we have left. Yeah. So we are discussing this question. So in this conversion, hmm, in this question, what they are saying, the conversion of atmospheric nitrogen to ammonia or nitrogenous compound is known as. So it means what is converting here? Nitrogen gas is converting to either ammonia or other nitrogenous compound. Okay, this is the process of we called as nitrogen fixation. Okay, everyone is saying about nitrification. What happened in nitrification? In nitrification, this ammonia is converted into different nitrogenous compounds, or you can say in nitrate forms specifically. These nitrate forms are mainly formed in the nitrification process. Okay, in nitrogen fixation, nitrogen is this specifically atmospheric nitrogen is converted. So for this question, answer is C. That's why when we study about the different microorganisms that we mainly used for the nitrogen fixation, so what those microorganisms do? They mainly take the nitrogen from the atmosphere and convert into the nitrogenous compound that can be used directly by the plants. Okay. So for this question, answer is C. So everyone understood this question and others also. If there is any issue in any question, then please let me know. Okay, someone asked a question. Ma'am, can you explain 13th question again? Yeah, just a minute. I'll share one figure with you. My screen is visible like another screen.
Yes, ma'am, it's visible.
Yeah, just a minute. Are you able to see the picture clearly or not? This is yeah. Now this is correct. You can see when this atmospheric nitrogen is converted to nitrogen fixing, like means when the conversion of this nitrogen, atmospheric nitrogen is done, this process is called as nitrogen fixation. Okay. Suppose you can see in the last step where this ammonia is forming, or this nitrogen is used by the nitrogen fixing bacteria. So this is mainly the nitrogen fixation. Okay, but when this ammonium, you can see now the change, when this ammonium is changed to nitrates or the other nitrates compound, this process is called as nitrification. Now understood the difference, that when ammonium is converted to nitrates and other nitrates, then it will form this process is called as nitrification. And when this ammonium, atmospheric nitrogen, is converted to ammonia, this is the nitrogen fixation. Now understood the difference between these two processes? Anyone has any other doubt in any question? Okay, Satvika, you are asking 13th question. Again. Oh, okay. This one you are asking.
Yes, ma'am.
Okay, I'll explain it. So what they are asking, they are saying the following is our example of microorganism used for phosphate fertilizer. Okay. Options given are Pseudomonas, Aspergillus, Mycorrhiza, and the option is all of the above. So what happened? What is mean by phosphate fertilizer? Normally, why we provide the fertilizer? So that it will give the important component to the plant for their growth. So phosphate fertilizer means we are providing the phosphorus to the plant. Okay. In this table or figure, you can see that here are the examples of different types of fertilizers. So in the second one, you can see this is phosphorus solubilizing biofertilizer, and third one they have given phosphate mobilizing fertilizer. So what is the major function of these two type of biofertilizers? It means they provide the phosphorus to the plant, but both of have their different pathways. Okay, it means the this PSB, phosphorus solubilizing biofertilizer, they provide the phosphorus to the plant in a different way by making it soluble, not exactly soluble, but like in a converted form so that they can directly use this. And this th- third type, phosphate mobilizing biofertilizer, for this example is mycorrhiza, they will also provide the phosphorus to the plant. This is phosphorus, okay. But the working of this biofertilizer or these microorganisms is different. Like one is, okay, what they will do, first one, it will convert it inorganic and organic compound to the phosphorus, and this mobilizing what it will form, that suppose the phosphate phosphorus is present in a different form, then it will make available that it can be used directly by the plant. So both these are providing phosphorus to the plant. So both these are example of phosphate fertilizer. Okay. So examples are Bacillus, Pseudomonas, Aspergillus, and Mycorrhiza. So in this question, options are Pseudomonas, Aspergillus, and Mycorrhiza. So all the three are used as a phosphate fertilizer. Now understood this question?
Yes, ma'am.
Okay. If anyone has any other doubt,
Ma'am, I have doubt in that quiz 10, ma'am. Actually, in question number 10, this was already like in the previous assignment you have told previous week.
oh, okay, yes.
So the question is like which one of the following, which is not a method of active draining techniques in cheese making process?
Yeah,
For that, the option they are given is stretching, kneading, cutting, and all of the above. But these all are the techniques, which is a method which we'll be using for the active draining technique. But in the question, they have asked not a method. But if you are answering for all of the above, then all of the methods which we are not using like that, it would be answered, ma'am.
sorry, what you have said at the last? Hello?
Ma'am, in question number 10, they have asked which is not a method of active draining technique.
Yeah, I understand.
Active draining techniques in cheese making process.
Yeah,
So the options were stretching, kneading, cutting, and all of the above.
Yeah.
So like if you are answering for any one of the options, like all of the options they are involved in the active cheese making process only.
Yeah,
Okay.
ma'am.
Active draining techniques in
Last time when I asked, you said whatever is nearest one, you choose that one.
Yeah.
So
Correct.
I did the same thing, but they have announced the assignment solutions. So in that,
if
they are saying
we
all
have
of the above.
answered one question but they have but it was showing not correct, but if you are
Okay.
answering for all of the above also like
They
if
are
you are
saying.
answering for that question it will be wrong only.
Okay. For that, you are saying both the options they are showing incorrect. All of the above also, and if you choose the one option from those three techniques.
No, if the question if they are asking what are the methods they are using for active draining technique, then for that if you are answering all of the above then it is correct.
Okay. Okay. You are
But
saying
in the question they are asking which is not a
not a
method.
method. Okay. Okay. I understood your problem. Okay.
We all should get mark.
So for that, if you are answering all of the above, then all the three process which is not involving like that, it is coming, na the meaning.
Yeah. Yeah. Okay. I understood. That is probably the printing error. Okay. I'll mail them. I can only do this thing. Like I can ask
Yeah,
them this.
because we have got a wrong answer for this. Actually, I have got a wrong answer for this. So
Yeah.
kindly see that, na man. Like,
Okay. Okay. Yeah. I understood your issue. Okay. Any
yeah,
other
thank you,
question?
ma'am. Kindly do the needful, ma'am. Thank you, ma'am.
Thank you. Okay. If there is no doubt, then we will meet next week. And after that, you all have your exam for the same.